In this session, we will discuss about the limitations of Beer's-Lambert's law. Basically, we will be trying to understand the law, the limitations when this law is not obeyed. So, this law, the Beer's-Lambert's law, is not obeyed when, in the first case, when different forms of absorbing molecules are in equilibrium, as in keto-enol tautomers. Basically, what this means is when I have an absorbing species, if that absorbing species or chemical entity, if it is going around some chemical changes, or if it is a keto form, if it is in equilibration with the enol form, or the enol form is vice versa to a keto form, then basically what happens is the structure keeps on changing, and the electronic parameters will also keep on changing with the structure. So if there is change in the electronic parameters, then definitely the absorption pattern for both the chemical entities, that is keto and enol form, will be different. Okay, so if that is the case, if any chemical entity, if it is undergoing equilibration between the two chemical forms, then basically that entity will not obey the Beer's-Lambert's law. And the second case when it is not obeyed is when the fluorescent compounds are present. So basically, what happens with the fluorescent compounds is these fluorescent compounds will have altogether a different electronic excitation and relaxation process. So if that fluorescent compound is having a different relaxation process, I mean to say, when the electron is excited, if it is not shifting to the ground state but rather going through some other state, then coming back to the ground state, that is also accounted during the absorption process. So basically, the fluorescent compounds will not obey the Beer's-Lambert's law because of the different electronic excitation and relaxation processes. When solute and solvent form complexes through some sort of association, so this is also very crucial. The solute what you have taken and the solvent what you have taken, in order to dissolve the solute, that solvent should not have any sort of chemical interactions with the solute. Basically, if the solvent is interacting with the solute and is forming some bonds, or is associating in the form of chemical bonds, so then what that ultimately results in the difference in the structure of the solute. If that is happening, then if there is a formation of complex, then that complex behaves as a separate entity and the solute behaves as a separate entity. Then it is like having two different compounds which are having altogether a different nature. So that is a form sort of association which we don't expect in the limitations of Beer's-Lambert's law. So the next thing is, if the structure of the colored ion or the colored non-electrolyte in the dissolved state changes with concentration, then that is also disobeyed in the Beer's-Lambert's law. I mean to say, if the structure of the colored ion or the colored non-electrolyte in the dissolved state, if it changes with the concentration, there are some compounds of whose the color changes with the concentration. Basically, if the concentration is higher, the color of the same compound will be different. If the concentration of the same compound, if it is very low, then the color is different. So basically, the structure of the colored ion, it is very much dependent on the dissolved state, and if it is changing with the concentration, then the Beer's-Lambert's law will not be applicable. So the next point is, if the colored solute ionizes or dissociates or associates in the solution, example the dichromate ion, which changes the color changes on dilution. So basically, it is somewhat similar to a sort of the previous statement, where the structure or the color, if it is changing, then the Beer's-Lambert's law will not be applicable. And the next important thing is, if the monochromatic light is not used. I have already said what we mean by monochromatic light. Monochromatic light is a light which is comprising of a defined set of wavelengths, or a set of wavelengths which are lying in a defined wavelength region. If we are not using those monochromatic lights, then definitely the Beer's-Lambert's law will not be obeyed. If the width of the slit is not proper, therefore allowing the stray radiation to fall on the detector. So in the instrumentation part, there is a source of radiation, there is a slit, there is a filter, there is a wavelength selector, then there is a sample cell, then there is a detector. So if you don't maintain the slit width, which is allowing a proper amount of incoming radiations, if you don't maintain the width of slit, what happens is basically some amount of radiation will be left out to fall on the detector, and that radiation will be actually interfering with the absorption, reading of the absorption. Basically, that is also one more limitation of this Beer's-Lambert's law. And one more is, if the solution species undergo polymerization, it is a sort of one chemical entity getting along with other chemical entity, and they are forming a sort of bonds, and they are forming a polymerization process. Then that actually that whole polymerized compound does not behave as an individual compound. Rather, it behaves as one particular moiety which is having many individual units. So that is the process where small individual units come along and polymerize themselves to form something very big. Okay, if that is happening in this particular case, or if there is a polymerization process that is happening, then the Beer's-Lambert's law cannot be applied in that particular process. We have one example for this polymerization process, that is benzyl alcohol in carbon tetrachloride in high concentration exists in polymeric form. Dissociation of this polymer increases on the dilution. I mean to say, it is a sort of polymerization, then the dissociation of the polymer, later on dilution. So that is how this benzyl alcohol in carbon tetrachloride cannot be measured, or it won't obey the Beer's-Lambert's law. So these are the cases in which the Beer's-Lambert's law will not be obeyed. Coming to the next case, when the sample is of high concentration, at higher concentrations the individual particles of an halide no longer behave independently of one another. Absorptivity A and the molar absorptivity epsilon depend on the sample's refractive index. Since the refractive index varies with an halide concentration, the value of A and epsilon will change. I mean to say, when we have a sample in very high concentration, then both the absorptivity and the absorbance will definitely change. Okay, so the compound will behave very differently in the higher concentration rather than in the lower concentration. So I have divided whole of the limitations into two sets now. The one is the instrumentation limitations, the other one is the limitations related to the sample. The instrumental limitations, what we say was the stray light. If it is any stray light which is coming out of the source and falling on the detector, which is not needed, if it is falling on the detector, then that may disturb the absorbance pattern. And signal to noise ratio is one of the important concepts, wherein which we need to differentiate the signal from the background noise. Okay, so that is also considered, and certain ratio is fixed in order to have a particular signal which can be actually taken out from the background noise, where a signal can be identified or a peak of sufficient height is excluded, so that it can be accounted as a signal. Coming to the resolution, that is also one of the instrument limitation. Resolution is something where the two closely lying things should be differentiated properly. If they are not differentiated properly, then we say it is of poor resolution. So these are the things which are with respect to the instrument side. And with respect to the sample, we have some of the limitations which we have discussed in the previous slide. The molecular interactions, associations and dissociations, dilution, those all are the molecular interactions. Hydrogen bonding, yes, that is also an important parameter which is affecting the absorption pattern. Reactions, if any reaction, if a compound is undergoing a reaction and it forms a product during the course of observation, then what happens is the compound is no more a compound, but rather it is a product or the degradant of that particular compound which is being measured. If that is undergoing any reaction, then that compound will not behave as an individual entity, but it is the mixture of things which appear as a peak in the UV spectroscopy. So refractive index changes, which we just now studied here. The refractive index changes with the concentration, and the concentration is directly dependent on the absorptivity as well as the absorbance. So within the Beer's range, the absorptivity will never change, but if we exceed or if we cross that Beer's range, the epsilon, that is molar absorptivity, will definitely change. So these were some of the limitations what we had in the Beer's-Lambert's law. So coming to the next part, which is the core concept of this electronic spectroscopy, where we are supposed to understand the type of orbitals we have in any given molecule, so that the transition between these orbitals, the study of transition between these orbitals becomes very important in this UV spectroscopy. So in this electronic transitions, we need to understand what we mean by molecular orbital, what we mean by sigma orbital, what we mean by pi orbital, and what we mean by electrons. So molecular orbital is something, or it is a non-localized field between the atoms that are occupied by bonding electrons. I mean to say, these are non-localized fields between the atoms where the electron can occupy its space. When two atom orbitals combine, either a lower energy bonding molecular orbital or a higher energy anti-bonding molecular orbital may arise. So that is basically when molecular orbitals combine, a lower energy bonding orbital, bonding molecular orbital, or the higher energy anti-bonding molecular orbitals may result. And the next kind of orbitals are the sigma orbitals. The molecular orbitals associated with single bonds in organic compounds, they are called as sigma orbitals. And with respect to the pi orbitals, the molecular orbitals associated with the parallel overlap of p orbitals or atomic p orbitals, we call them as the pi orbitals. And the n electrons are something where these electrons are not involved in any bonding process. So with this preamble of molecular orbital, sigma orbitals, pi orbitals, and n orbital, we will study the concept of electronic transitions in UV spectroscopy. The electronic transitions are very much important and they should be understood if you are to understand the electronic spectroscopy. So on absorption of an electromagnetic radiation in the form or energy in the form of electromagnetic radiation by a molecule, the changes are evidently happening in the electronic energy states of the molecule due to the transitions of the valence electrons. There is some change that is observed in the electronic energy of the given molecule. So when a molecule is subjected to an electromagnetic or incoming electromagnetic radiations, that molecule will sense this energy and the valence electrons will take up that energy and they will basically move to various energy states. So the transitions, the electronic transitions consist of excitation of an electron from an occupied molecular orbital, that is a non-bonding p orbital or a bonding pi orbital. Here you can see there are three different kinds of orbitals. The first one is the bonding orbitals, and the second one is a non-bonding orbital, and the third one is the anti-bonding orbitals. Basically, if the transition of an electron, it usually happens from the bonding molecular orbital or a non-bonding orbital to an anti-bonding orbital, or the anti-bonding orbitals are usually said to be at a higher energy states. With this concept, we have four different types of transitions coming into picture. I have sigma bonds, I have pi bonds, and I have n electrons, which are sensing the energy change and they are taking up the energy and they are jumping to the higher energy or the anti-bonding orbital, that is pi star and the sigma star. So on accounting all these bonding, non-bonding and anti-bonding orbitals, we have four different kinds of transitions that are possible. We can see four different kinds of transitions that are